summarize my research that I have been doing during the past several years. So today I will be talking about my research using DNA landmark coding to assess beetle biodiversity for ecologically sustainable forest management. Okay, let's start with forests. Forests are very important as they provide ecological, social, and economic functions. For example, forests sustain huge biodiversity and regulates local and global climate. In addition, they provide educational and recreational functions. Last, they produce wood products and bioenergies. Well, unfortunately, the loss of forest is continuing. For example, the net loss of forest is around 5 million hectares per year during the past several decades. And given the essential services that forest provides to us, we need to make sure to manage forest in a sustainable way to ensure such services for our generation and future generations. Reconciling the biodiversity conservation and wood production is among one of the main topics. And actually, sustainably managing forests to halt biodiversity loss is among the Sustainable Development Goals of the United Nations. And Australia is one of the countries that have embedded the conservation of biodiversity into its framework for sustainable forest management. Putting ecologically sustainable forest management into the context of production forest, it is related to landscape planning. Recent research have gained knowledge about the importance of unharvested mature forest in maintaining key habitat elements as well as sustaining biodiversity of forest. Here is an image showing you what the unharvested mature forest looks like and how they are different from older regeneration forest and younger regeneration forest. My research will focus on old regeneration forest and I will use mature forest as reference. And here is a conceptual diagram showing you the importance of mature forest in the landscape. In the middle is a log size. And around its 1,000-meter radius buffer, there are 5 mature forest patches, and they provide key habitat elements and sustain source populations of forest. In addition, they also facilitate the biodiversity re-establishment in harvest areas in successional time. In order to understand the influence of mature forest on the biodiversity in harvest areas, we need to study the configuration of mature forest in the landscape. Here, I would like to introduce two main concepts. The first one is landscape context. It represents the percentage cover of mature forest around the harvest sites. And here again is the conceptual diagram. The total amount of mature forest in the example is 36%. The higher percentage of mature forest in the landscape, the stronger landscape context it is. And another concept is forest inference. It refers to the distance to a nearby mature forest. The forest inference in the example is 60 meters. The higher, the closer the unharvested mature forest to the logged area, the stronger the forest inference it provides. Landscape context provides source populations of forest species, while the forest inference facilitates the movement of forest species into harvested areas. And along with other configuration indices, they collectively influence the biodiversity recovery in harvested areas. So speaking of biodiversity, how do we measure them? Conventionally, we use indicator species. And beetles are very good indicator species due to their high diversity. And the diversity of beetles encapsulates the high species richness, trophic diversity, and beetles are also very sensitive to disturbance. So beetles are commonly used as indicator species in forest ecological studies. Then we look at our toolbox to see what tools do we have to study biodiversity, beetle biodiversity. Researchers have been using microscope to identify beetle species. It's very useful, but not very necessarily cost-effective. Actually, it's very time-consuming because it requires strong taxonomic expertise. And recently, a new method called DNA map barcoding has emerged. It's promising to assess biodiversity in a more cost-effective way and thus facilitate biodiversity assessment and forest management. Well, so far, I have been talking some backgrounds about forest. And here I would like to give you an overview of my talk, and they can be also decomposed into chapters of my thesis. So the first chapter is a general introduction, and the last bit is general discussion. And for DNA map barcoding, I have 3 chapters. The first is a review paper in which I recommend some guidelines for entomology. The second one is a comparison study where I compared DNA map barcoding with morphological identification. The third one is a study where I tested the quantitative performance of map barcoding. Chapter 5 is a landscape ecology study where I used DNA map barcoding to assess beetle biodiversity and examined the landscape drivers of beetle community in old regeneration forest. So, what is DNA map barcoding? This is DNA map barcoding explained in 60 seconds. Usually, we would have a DNA barcode database where photographs and the DNA sequences of species are deposited. It's analogous to product barcodes, where we can use a scanner to scan the barcode, and we will know what product it is. For DNA map barcoding, we usually start with multiple species, where we extract the DNA from the specimens. PCR amplification is used to generate million copies of DNA sequences for studied species. And in this case, DNA sequencer is used to scan the sequences. And with the help of DNA barcode database, we would know what species are they. And for species that doesn't have a reference in the database, they can be clustered into operational taxonomy units. They are more or less equivalent to species. I have published a review paper about DNA map barcoding, where I have recommended some guidelines for entomology. In that paper, I identified the key steps of a standard workflow of DNA map barcoding. I have recommended some considerations during field sampling. For example, I recommended some decontamination methods and also some DNA preservation methods. And also I have recommended some procedures for sample handling. I also identified some biases during laboratory processing step. Finally, I recommended some user-friendly interface programs for new users. DNA map barcoding is not a daunting tool as long as you have or you're willing to gain some wet lab skills and some bioinformatics. Having said that, if DNA map barcoding is to use as conventional approaches, we need to make sure it provides similar estimates of species composition and also provides similar conclusion. However, There is a few comparison studies compared the performance of DNA metabolic coding and morphologic identification in forest ecosystems. For example, in a study, invertebrate species composition between plantation and native forest are compared use DNA metabolic coding and morphologic identification. The ecological gradients are usually shown in these studies and can be easily detected. So again, if DNA metabolic coding is to use as a practical tool for biodiversity monitoring, we need to make sure it captures the strong ecological gradients, but as well as the subtle ones. So I set my comparison study in the context that subtle but significant differences of beetle community composition are expected between old regeneration forest and neighboring unharvested mature forest. I selected 12 pairs of sites in Tasmania Southern Forest. At each site, 7 traps were deployed to collect beetle specimens. Here again, I would like to remind you that the conditions in old regeneration forest are gradually recovering to that in unharvested mature forest. I used DNA metabolic coding and the microscope to identify beetle specimens. I ended up with around 1,900 individuals. They were identified to 173 species. The most species richness family is Staphylidae. The second species richness one is Cuculionidae. The third one is Leodidae. And there are many singleton species in many families. Then I compared the morphologic identification to the results of DNA metabolic coding. CO1 and 16S are 2 commonly used mitochondrial DNA markers. So metabolic coding of CO1 identified a similar number of species to morphologic identification, while metabolic coding of 16S identified a slightly less number of species. But when we look at each family, the number of identified species were sometimes not the same. For example, for Staphylidae, more species were identified by morphology, while, for example, for Cuculionidae, more species were identified by metabolic coding of 16S. And here I would like to emphasize that for DNA metabolic coding, the assignment of species name is really DNA barcode reference reliant. So for those without reference in that database, they can only be assigned to genus or family, or otherwise they are assigned to others in my study. And here are some results of species level comparison. We would expect 3 scenarios. The first one is correct identification, where species were identified by both methods. And the second one is false negative, where species were only detected by morphology, but not by metabolic coding. And the third one is false positives, where species only detected by metabolic coding, but not by morphology. And here is the results for CO1. Only 22 OTUs were assigned with species names, and it only a small portion of the total number of OTUs. And here in the table, each row represents a study site, and each column represents a study site, and each row represents the detection of a species across study sites. So in the table, the correct identifications, which are the blue dots dominate the results with a few false positives. I'd like to use the highlighter. And there are some false negatives randomly scattered into different species. And here is the 16S data set where we have 22 OTUs assigned with species names. And again, it only take up a small proportion of the total number of OTUs. And here, the general pattern was dominated by correct identification, which are the blue dots. And again, with a few false negatives. But for the false, positives, but for the false negatives, there are many occurred in a few species. And since only a small portion of OTUs were assigned with species names, I used the cross-test correlation to test the correlation between two data sets. The results showed significantly correlation between metabolic coding data sets with morphological data sets. And this suggests metabolic coding results is comparable to our morphological identification. And if we are aiming for a complete species name, definitely we need more reference DNA barcodes. And as noticed, there are a few false positives, and these are likely to be the detections where DNA metabolic coding to provide higher resolution. And here, I compared the beetle composition differences between regeneration forest and the mature forest. I plotted ordination using presence and absence datasets. And as we expected, the beetle's species composition are significantly different between mature size and regeneration size. The explained variance is pretty low, only 6%. And for CO1 dataset, a similar pattern was observed where the beetle species composition were significantly different between regeneration size and mature forest size. And the explained variance is slightly lower, suggesting some signal was lost. And this is likely due to the false negatives detection of some species. And for 16S, we didn't observe the similar pattern as in morphologic and CO1 datasets. And again, the reasons are the false negative detections, and as well as slightly less total number of OTUs were detected due to a poor reference database for 16S. So overall, metabolic coding identified the similar beetle composition as morphological identification does. And the selection of markers is important. So in my study, CO1 is a suitable marker for studying beetle species. And metabolic coding is promising to assess biodiversity because it offers a more cost-effective tool. And this study has been published in the journal Restoration Ecology. Well, so far I have been using presence and absence data for my analysis. So why not use species abundance data? Well, simply put, there is no research that have made this method reliable, generally reliable, but there is still potential that I want to explore. Here, I would like to give you some background of PCR. Yes, PCR chain reaction. So here we have the template DNA of a DNA marker for studied species, and we would like to use a pair of primers to amplify the template DNA, and we would expect perfect match between the primer and the binding region, where A and G, G and C's are perfect match. But what if we have one or more mismatches between the primer and binding regions? Well, chances are the amplification efficiency would drop, or there is no amplification at all. And here I would like to give you a more detailed example. And it shows the binding region of 100 beetle species for a given primer. On the x-axis is the size of the binding region, and for each row is a species. So for a given primer, it's really impossible to have a perfect match when you want to amplify the same DNA marker across 100 species. So in this case, in this study, I would like to understand the impacts of primer template mismatches on the quantitative metabolic coding. And again, further, I would like to ask how different number of primer template mismatches would affect species abundance estimation. And generally, we would expect three hypothetical outcomes for the quantitative DNA metabolic coding. For the first scenario, on the x-axis is the input biomass of a species, and on the y-axis is the output number of DNA sequences. So the first scenario is a perfect scenario where the output number of DNA sequences perfectly reflect the input biomass of species. And the second scenario shows some strong positive correlation between input biomass and output number of DNA sequences. And for the third scenario, there is no correlation at all, which is very useless. And previous studies have been using mock samples where only up to 50 or 60 species has been studied. But this study could hardly represent the complexity of real-world samples where more than 100 species were expected. So I used a data set from my last chapter where I have 22 species and 25 species identified for CO1 and the 16S marker respectively. And I'd like to emphasize that they are actually from real-world samples where 173 species were identified. So with that, I would like to ask, is there a correlation between species biomass and the number of DNA sequences for beetle species? And further, I would like to explore, is there a possibility to improve the species abundance estimation if there is a positive correlation? And is that possible to find some correction factors? And for the 22 species in the CO1 marker, the primal template mismatches range from 0 to 7, whereas the primal template mismatch were almost at 0 for 16S, means in most cases they are perfectly matched. So for CO1, the results only showed a marginal significance, but very little correlation between the biomass and the output number of DNA sequences. However, for 16S, there is a significant strong correlation between the input biomass and the output number of DNA sequences. And further, I would like to explore whether the deviation of the linear model has some taxonomic bias. So I extracted the residues from the linear model, and I mapped the deviation of sequence abundance into a phylogenetic tree. So I constructed a phylogenetic tree of the studied species, and I mapped the deviation of species abundance as well as the primer template mismatches onto the phylogenetic tree. So on the graph, there is a correlation where for species that have less primer template mismatches would have more DNA sequences than expected. And for species that have more primer template mismatches, they would have less DNA sequences than expected. But there is no such pattern observed for 16S dataset, because overall, the primer template mismatches is 0. So the DNA sequences were overall evenly represented across studied species. So to summarize, quantitative metabolic coding has some promise, but it's not ready yet. And second, whether to use CO1 or 16S, my results showed that 16S has shows better correlation between biomass and output number of DNA sequences. But for CO1, it has some advantages in bioinformatics analysis, and it has more comprehensive database. But it doesn't mean that 16S is less useful. So if we can build up a more comprehensive database for 16S, it might have a comparable results to CO1. And in my study, I only looked at primer template mismatches, but there are some other factors impacting quantitative metabolic coding. For example, the positions of primer template mismatches, the efficiency of DNA polymerase. And I would like to further validate my findings with more established data sets in the future, because in my data set, due to the limited reference database, only 22 and 25 species were identified for 2 markers. So overall, my studies has demonstrated that DNA metabolic coding is a useful tool for beetle biodiversity assessing. So I will use DNA metabolic coding for a landscape ecology study. Here, I would like to give you a refresh where I look at the landscape configuration of mature forest. Previous study have found forest inference has positive influence on the biodiversity recovery in harvested areas, and the same applies to landscape context. But in my study, I would like to explore the interactions between forest inference and landscape context. And also, I would like to examine the landscape drivers of beetle community composition in old regeneration forest. So I selected my field size in old regeneration forest, which I selected sites represent both high and low level of landscape context and high and low level of forest influence. So for each class, I have selected 16 sites, and they can be further randomly scattered into 16 classes, as you can see in this graph. And here I compared the beetle species composition between regeneration forest and mature forest. And here is a ordination constrained by forest class. And the yellow ellipse represents the mature forest size. So in this ordination, there is a clear separation between regeneration size and mature forest size. And second, the classes of regeneration forest that have more landscape context, which are the black ellipse and the yellow ellipse, have slightly more overlap with the mature forest. And there is no evidence for sites with great forest influence being closer to mature forest. And this is the same for smaller 125-meter distance classes. And the results are not shown here. And further, I would like to examine the landscape drivers of the beetle community composition. So I selected 5 categories of explanatory variables. The first category is spatial variables, where I produced a bunch of principal coordinates of neighborhood matrices. There are 42 PCNMs, where PCNM1 represents cause scale variation, and PCNM42 represents fine scale variation. The second category is topographic variables. I looked at elevation, topographic position index, slope, eastness, northness, and third is bioinformatic, where annual chance, seasonality, and extreme environment factors were considered. And the fourth category are landscape configuration, which I mainly look at forest influence and the landscape context, along with some other matrices related to patch shape. And the last one is regeneration age. So I used generalized dissimilarity modeling to examine to study their relative importance. So the overall explained variance was 34%, and the spatial variables account for most of the relative importance of the beetle composition dissimilarity. And the second one is topographic variable. And here I showed you the top two important variables for each category. And on the x-axis is the gradient of the variable, and the height of the y-axis is the magnitude of the beetle species composition account by each variable. So the PCNM 37 and 42 respectively account for 9% and 5% of the relative importance of beetle species composition. And for topographic variables, the topographic position index and elevation account for most of the variance. Both the topographic position index and the elevation, they influence the movement of beetles in the landscape. And for bioclimatic variables, the precipitation of driest month and mean temperature of wettest quarter were identified as the most important bioclimatic variables. And when we look at the most important landscape configuration indexes, which are the landscape context and the forest influence, although they only account 2% and 1% of the relative importance, we identified a threshold where 25% of the landscape context, when it's beyond 25%, the beetle species composition were mostly accounted, mostly explained. And while for forest influence, there is a threshold, the curve reached a plateau at around 200 meters. And this suggests that forest influence would be more important along over shorter distances. And this is in line with some previous findings where the distance effects of mature forest reached a 176 meter. And with that, I conclude that I have some recommendations to forest managers where I recommend to the consideration of local scale habitat heterogeneity, where my results, the PCNMs and topographic variables explained most of the local scale variations for beetle species composition. And here, the regeneration age also explained some relative importance, suggesting having a mix of age class could also help the regeneration forest to sustain their own biodiversity. And last, maintaining a certain amount of mature forest is important in the landscape, as my results showed. And also, I would like to summarize the pros and cons for DNA metabolic coding. So, if our aim is to get a complete species name, probably we still need to build up more comprehensive reference database. And if the aim is to estimate species abundance, there is still some potential to improve the accuracy. And as an emerging approach, there are some challenges around bioinformatics and also some genetics skills for new users. But my study have demonstrated that studying species composition with DNA metabolic coding is comparable as morphological identification, and its advantages of DNA metabolic coding for scaling up to study multi-taxa and for large scale biodiversity. And some thoughts on future research. My study have only focused on beetle species, while some research have suggested that different taxonomic groups would respond differently to forest practices. So future research will also need to look at impacts on other taxonomic groups, such as vegetation and birds. And also my study sites were generation forest following first rotation. And it's still unknown whether the effects of mature forest could also last for regeneration forest following multiple harvest rotations. And last, if there's still potential for using DNA metabolism to study species abundance. And with that, I would like to thank you many people. First of all, I would like to thank my supervisors, Chris Burridge, Sue Baker, Greg Jordan, Lawrence Clark. They have been very supportive and in my research, as well as for my career development, I'm very grateful to. And also I would like to thank to Lynn Foster, who has been helping with my morphological identification, and Sam Wood for helping with GIS, and Marie and Rob from STTES with my study design, and Simon Grove from TMAC, who borrowed some beetle specimens. And also I'd like to thank some people who helped with genetics and bioinformatics. Florian from CSIRO, James Mathick from Menzies, and thank you for Valerie for allowing me to use the PC2 lab, and also Adam, Sherry from CSL. They have been kindly help with my genetics work, and a lot of wonderful, amazing volunteers with my during my field work. I couldn't finish my field work, get my data without them. So I really grateful to them. And last are our amazing administration officers, Morgan Green, Kath, Sophie, Shelley, and Anna. They have been always very helpful, and I thank them. And special thanks to my wife, Dian Fan. And as Sue mentioned, that we got married earlier this year in China, but currently we are separated. By finishing my talk, I would like to express a message to her. If I can finish my PhD, there is no way you cannot. And I think you can hear me, because probably you are in the audience, and I really miss you. Finally, thank you again for your attention. I would like to take some questions.
Okay, thanks Mingxin for a great talk. While I think of it, Mingxin mentioned before we started that he'll be going down for a drink at Preachers after the seminar, and everyone's welcome to come and join him and celebrate. It's also really the end of his PhD, because he'll be handing in very soon. So now for questions. Right, so I can see that Hans has his hand up. So over to you, Hans.
Is that working?
Yes.
Cool. Yeah, well done, Mingxin. That was really cool, very comprehensive.
Thank you.
Covered a lot of ground. I had a couple of just questions on some of the bioinformatics components. kind of more like speculation, I suppose. But regarding the reference database that you use to get your taxonomies, if that is quite incomplete, what percentage sequence matching are you using to call your taxonomies on the OTUs?
Are you asking the length of the DNA marker, or?
Oh, the length of the I guess sequence matches that you use to accept it as a taxonomy?
Yeah, I use the UNOIS 3 algorithms where I match 100% to the OTUs.
Oh, okay, well.
Actually, I use zero radius OTUs, so, and I use 100% match.
Oh, okay, and so when you take those zero radius OTUs and put them against the reference database, what percentage identity do you accept to call it as that's its taxonomy?
Yeah, I think I haven't explained myself clear. Like with usearch, I mean the zero radius OTUs, first I class them into ZOTUs, then I used a percentage of 100% to match the sequences against the ZOTUs.
Okay, interesting.
Yeah.
That's cool. My other one was just, it was quite interesting how you got a great relationship between the input DNA abundance and the output of the reads for your 16S amplicon. Because notoriously 16S exists in multiple copies in bacteria, sometimes certain species will have up to 10 copies. Is that, I guess it's indicative in your graph that that isn't the case in beetles, but do you know much more about the number of 16S genes within beetles?
Yeah, like as I mentioned, like there are other factors could impact the quantitative metabolic coding. One of the reasons could be the copy of the mitochondrial DNA, which you know they might have like different species, beetle species might have different copy number of mitochondria. So I think that's also a reason, like a factor need to be considered if we want to improve the accuracy of species abundance. But for my study, I haven't considered that because I only look at the like the number of primal template mismatches. But yeah, that's a good point.
Cool. Okay. Yeah. Thanks, Mingxin. Awesome. Great stuff.
Okay. So Leon.
Thanks, Sue. Thank you, Mingxin. That was really interesting. I have so many questions, and I wish I could come to preachers, but I'll limit myself to one. The large contribution of the spatial component in your analyses. Why do you think that is? Is it because there's a lot of biogeographic diversity underneath all this? In other words, beetles are fairly poor at dispersing, and so different parts of the forest estate are actually got really quite different species of high beta diversity, or is there something else going on?
Yeah, like when I have the results, I had to think about why the PCNMs are account for most of the species composition dissimilarities. And I think the reasons are I'm looking at regeneration forests after around 50 years old, and I think they gradually recovered and sustain their own biodiversity, and there are some microhabitat variables that I didn't consider in my study. So I think those variables, those microhabitat variables, could explain why most of the beetle species dissimilarities were accounted by local scale PCNMs. I'm not sure that's...
Yeah, it's sort of, yes. I'm just wondering whether there might be, obviously you don't have time to look at this in your PhD, but if people have thought about looking at trait-based versions of these analyses for forest beetles to see whether some of the noise that might be induced by sort of having very highly localized species distributions might be sort of fuzzed out, and you might get a clearer signal in functional terms of what the forest type is doing. Yeah.
Yeah, indeed. You raised a very good point. Yeah, I came across like a couple of studies. They also include some like function traits, for example, like how the length of the body, and some beetle species. They even though I look at ground active beetles, but some species they might have more. I mean, locomotion ability than others. Yeah, but definitely, yeah, that's very a good one.
Thank you.
Tim Wardle, I think, has a question.
Thanks, Liz. Mingxin, well done you, getting this far with all that upheaval in your life.
Yeah, thank you.
Like all the other questioners, there are a lot of things I'd like to ask you, and I don't know where to start. So I think the one I'd like to ask is the role of very rare, or the dominant role, is it the very rare or the very uncommon species in contributing to your results? Is that, were they excluded in your analysis, or did you look at all your species in trying to untangle the ecological effect between your sites, or did you just look at the species?
Sorry, I didn't quite follow your question. Can you say it again, Tim?
Okay, Mingxin, did you so yeah, keep going back. Back.
Is this one?
Yeah, okay, that'll do. When you look at when you've done the ordinations, with all the species you collected, or did you filter it and just do the ordinations with your most common species?
No, that's all species. Yeah, that's all species. I didn't filter the rare species. But there is some bioinformatic analysis that might affect the total number of OTUs, because I use OTUs not because I can't get a complete list of species. So that's like the data sets of OTUs, which I have demonstrated in a previous study that they are equivalent to capture the species composition as morphological method.
Thank you.
Does anyone have one last question?